The next structures we have are the lysosomes and peroxisomes, and as I said already, these are particular structures that are useful in the degradation of specific components that come into the cell. Lysosomes in particular will degrade waste products generated by the cell, and these particularly originate in the Golgi complex, and they consist of enzymes that are called lysozymes that are used to break down specific waste products. Peroxisomes are interesting structures because they are structures that rely or that are useful in breaking down invasive components that come into a cell. So something such as a virus, maybe, or something like a bacterium that can actually pass through your cell membrane and find itself inside of the cytoplasm of the cell. Once it comes into contact with a peroxisome, it will fuse with the peroxisome, and then the peroxisome will release hydrogen peroxide onto that specific invasive entity or agent and start to break it apart. And then once it's broken apart, what will happen is the peroxisome will release those waste products outside of the cell. So in this diagram right here, this is a neat figure that highlights what happens when you have these peroxisomes and these lysosomes taking effect on waste components. So here's a Golgi apparatus. You have these lysosomes being released. The lysosomes are coming into contact with something that's called a food vacuole, and this is simply a food particle that's been ingested by the cell through the process of endocytosis. So it's fused in through the membrane and now has part of the membrane surrounding it. The membrane will then fuse with the lysosome, and the lysosome will start releasing enzymes to break down the food particles that exist in this vacuole. And so the vacuole becomes essentially a stomach or a digestive vacuole to break down those components, and those components are extracted and used by the cell, with waste products being sent out of the cell through the process of exocytosis. So that's how lysosomes and peroxisomes work. And peroxisomes do the same thing. They just are looking for anything that's foreign that shouldn't be inside of the cell, but they also work through the process of endocytosis and exocytosis. So these are some pretty neat structures in my opinion. In the more advanced microbiology courses, we dive into great detail on how these processes take place. The next structure we have is that of the mitochondria. I'm not going to spend too much time on these guys because these are kind of, this is kind of biology, high school biology here. So you should have gotten this at some point in your academic career in high school. But the mitochondria is where energy is generated in the cell, and we spend a lot of time talking about the process of glycolysis, the Krebs cycle, and then the process of the electron transport chain. So remember, in the process of cell respiration, you have glycolysis that takes place where you have the conversion of a glucose molecule into 3 sugar molecules known as pyruvate. And then that pyruvate goes through the process of the citric acid cycle, which is also known as the Krebs cycle. And then from the Krebs cycle, we have the electron transport chain. And then at the end of that, you have the generation of AT molecules that can be used by the cell in order to undergo DNA replication, undergo messenger RNA transcription, and then protein translation. Now, the number of mitochondria that exist inside of a cell depends on what type of cell you have. So like in our bodies, you have cells, that are fit for one specific function, such as lining the esophagus or those that line the trachea. But depending on, what that cell is functioning for, that will determine how many powerhouses of mitochondria are going to exist inside of the cell. And here are some pictograms from microscopes where you can easily see some of these membranes that exist inside of the mitochondria, and that's kind of the typical function or typical structure that you'll see if you observe mitochondria underneath the microscope. Mitochondria are kind of interesting because they represent, or at least they're kind of similar to what we think of when we think about very early prokaryotic organisms. They contain their own cytoplasm, they contain a cellular envelope, and then there's also DNA that exists inside the mitochondria. So there's a nucleoid region that exists throughout. So because mitochondria have their own DNA, there's a theory that's kind of thought, you know, of how prokaryotic cells originally were the precursors to the eukaryotic cells. Some theorems say that mitochondria used to operate independently as their own organism because they have their own genetic material. So in a sense, the mitochondria was basically a bacterial cell. Well, then you had a larger cell that had a nuclear membrane-bound nucleus that didn't have any mitochondria, ingested this mitochondria that was living as a bacterium, likely. And through that process of ingestion, it just didn't break down the mitochondria once it was absorbed. And so the end result was the fact that you ended up with a cell that had mitochondria living inside of it through a very symbiotic relationship where the organisms were not harmed. So you can think of these mitochondria as once being prokaryotic cells that lived all by themselves, and then they were ingested by a giant eukaryotic cell, and then they started living and residing inside the eukaryotic cell and serving the functions of the eukaryotic cell in order to survive. So that's just kind of a theory in terms of the origins of life that's out there if you never got that anywhere in your academic educational background. The next structures that we'll talk about are going to be that of plastids. Plastids are also comprised of a membrane, and these are going to be primarily found in photosynthetic organisms like algae or plant cells. We know this because these plastids consist of some sort of photosynthetic pigment where photolight is going to be trapped by the pigments that are incorporated in the membranes of these plastids. So we have things like etioplasts, proplastids, chloroplasts, I'm sure you've heard of. This is where the process of photosynthesis takes place. There's also leucoplasts, chromoplasts, amyloplasts, protein protoplasts, statolith. There's lots of different plastids that are out there. Photosynthesis is going to take place in a number of these plastids that exist, and the most common one that we know about is that of the chloroplasts, because the chloroplasts contain chlorophyll, and that chlorophyll gets excited when it comes into contact with electrons absorbed from sunlight, and then that sunlight is used to generate and produce glucose and sucrose molecules for a plant. Some cells are made up of a structure called a cytoskeleton, and the cytoskeleton is simply a complex system of fibers that are found throughout the cytoplasm. And the cytoskeleton just helps, again, to hold everything in place. The fibers that make up this system are known as microtubules, microfilaments, and intermediate filaments. So you can think of these fibers as little threads that produce this skeleton in which you have organelles residing. So this makes up the bulk of the cytoplasm and also provides structure to the cytoplasm itself. All these microtubules and microfilaments, they play a significant role in assisting all of the activities that take place in the cytoskeleton. The cell wall is another structure that can be found when we talk about cells. It's not common to all cells. In fact, there are things like animal cells that do not have a cell wall. But organisms such as bacteria, organisms such as fungus, organisms like plants, they all contain a cell wall. Why animal cells don't have this wall anymore hasn't really been the reason for that hasn't really been found out yet. But somewhere in the process of evolution, animal cells decided to get rid of the cell wall. Some eukaryotic cells have this wall, as I said. The wall is used to provide shape, protection, and structure for the cell itself. So it's an additional backbone, really, to providing support and protection. This particular cell wall that you see here in this figure is the cell wall of a plant. And we know that because you can see these chloroplasts that are inside of here. Chloroplasts, people get them very mixed up with mitochondria. Plants have mitochondria to generate ATP, but they also have these chloroplasts to absorb light and undergo the process of photosynthesis. So when you look at, if I gave you a picture of it, like of this cell on a test and ask you if it's an animal, plant cell, or bacterial cell, you definitely should be able to tell me that this is a plant cell because of the presence of chloroplasts, which are these green structures here. Okay. So the prokaryotic cells also, like I said, have a cell wall to them. The cell wall is kind of special because it contains specific types of proteins that make it up. And one protein in particular is referred to as chitin, which you can find in the cell walls of fungal organisms. Now, in the presence of algae cells and plant cells, we actually find a different component that's known as cellulose in the cell walls. And this is kind of important to understand what the cell wall is made out of, because the cell wall, not only does it provide protection for the cell, but it also kind of inhibits larger complex organisms from ingesting that particular group of organisms. So things like cellulose, humans have a very hard time digesting the cellulose that's embedded inside of this cell wall. So our bodies have to be equipped with special enzymes as well as special bacteria that can help us to break down the components of the cell wall. Okay, so let's talk about some comparisons between plant cells and bacterial cells. And bacterial cells, remember we talked about, well, I guess we haven't talked too much about the structure of their cell membrane, but I know in previous lectures I have referred to bacterial cells as having, or as being gram-negative or gram-positive. And that means that the structure surrounding a bacterial cell, the structure of the cell wall or cell membrane, is completely different from what you're going to see on a plant cell or an animal cell. So let's look at the plant cell first. And the only thing I really want you to take away from this particular diagram is the fact that you have this plant cell wall that consists of these kind of different sections. We have the middle lamella. We have the middle primary cell wall, and then we have a plasma membrane that surrounds the cytoplasm. Okay. The structures that make up the primary cell wall consist of certain molecules and protein structures known as pectin and glycan and microfibrils. So you can think of all of these different structures as different types of Lego blocks. So pectin, think of it as a chain or round Lego block. The cross-linking glycan, these are these green structures that you see throughout here. That's essentially sugar molecules called glycan that are joined together, providing stability for this primary cell wall structure. And then we have the microfibrils, which are made out of cellulose that I told you, remember, our cells typically can't break down when we digest them, that make up the microfibrils. And so this complex here really makes up the bulk work of what provides the protection and support and stability of a plant cell wall structure. And so all of that is situated above the plasma membrane, so it prevents items from causing the cell membrane or the plasma membrane to collapse on itself.